I want to thank the organizers for having me here and giving me a chance to present some of our work. And when I say our work, I'm presenting on behalf of the folks who really did the work, Najib Majaj, a postdoc in the lab, Ha Hung, who's here and presented a poster this afternoon, Ethan Solomon, an undergraduate, and Nicholas Pinto, a recent graduate student. So many of you who know me know my lab is interested in the problem of object recognition in the brain. And I've tried to put together something today that I haven't presented before, but I think is resonant with the population coding goals of this workshop. So again, it's all unpublished data that I'm going to show you for the second half of the talk. So I'm looking forward to feedback there. I start a lot of my talks by reminding folks of part of the reason that we've got into this business is because we think that engineers could use some help from neuroscience, that is, understanding tricks from the brain that can help build machines. And I like to remind people on the problem of object recognition. In 1966, MIT put together a summer project, and I thought this was apocryphal until Mike Tarr sent me this, that they planned to essentially solve pattern recognition in a summer, getting a couple undergraduate students together. They had sub-goals for July and August. Needless to say, that hasn't worked out, and we're about 60 years later, and so this is still quite a difficult and unsolved problem. So I'd also like to preface by saying, what is object recognition? We were talking about this at dinner last night. I want to remind you that object recognition is not all of vision. Take this scene. You might be able to navigate this scene, decide if this person is friendly. You could ask lots of questions about this scene. But we think it's a gateway into the general problem of scene understanding and perhaps cortical computation in general. And I could ask you a question about this scene, like label all the objects. And you might come back at me with something like this. You'd put labels and you draw boxes around things. And so this starts to look like standard computer vision. And that's the framework in which we think there's already been a lot of work. So we like to try to work within that framework as a gateway to the larger problem. So I want to also set the time scale here, so in how we analyze things in our data and think about the part of the problem that we're working on. So when you explore a scene, this is from David Scheinberg's lab. This is monkeys exploring a scene. You make lots of eye movements. You fixate for about 200 milliseconds at each particular location. Then you make rapid eye movements. So about 5 eye movements per second in monkeys and comparable, a little bit slower in humans. And so that's the time scale of interest. The spatial scale of interest of our lab is about the central 10 degrees of visual field. This is the portion of the visual field that essentially the ventral stream up to IT is really focused on analyzing. So that sort of sets the scale of things here. And now we come back to this full scene. The way we think about this scene being digested is you don't digest it all at once, but of course you make rapid eye movements around the scene. And what's actually hitting your sort of ventral stream engine, if you will, as I just described it, is something that looks a little more like this. So a series of snapshots there. And you might have noticed that was about 5 per second. You might have noticed you could see one or more objects in each and every frame. You saw a car, a person, a sign. That's kind of how I view the ventral stream as digesting at that time scale and that spatial scale. So here's another way to illustrate that. So we call this ability, this sort of engine of the ventral stream, we call this core recognition or core object detection. When you watch this movie that I like to show a lot, this is 6 frames per second. You notice you can sort of name, or that is, recognize one or more objects, label one or more objects in each and every one of these images, again, even though quite fast. Recognition is very easy for us. It's fast, feels effortless. I don't have to pre-cue you. I don't need to bring attention to bear in a strong way. I don't have to tell you what objects you might see. I didn't have to say anything about Leaning Towers of Pisa or Yoda, yet somehow you simultaneously can consider many of these possibilities in parallel at this very fast rate. It's most importantly, computationally, object recognition has been pointed out by several folks, is highly tolerant to variation, and this turns out to be the crux problem, so I'll talk a lot about that. And I'm also going to say that current machines, we think, are not yet near the level at which humans can do this, and I will show you some data to support that argument. The mission of my lab, then, is to try to understand how the brain can build a neuronal representation that can support these kind of, what I'm calling core recognition, not all of vision, but these kind of tasks. And again, we think of that as a gateway problem. And our operational goal, when we say understand, we really mean to build or discover brain-constrained algorithms that we can build into hardware that work as well as behavior. So that's our operational measure of how well we're succeeding at this goal of understanding. So let me sort of also say that my lab is primarily a systems neuroscience lab, but we also have folks that work more in the field of computer vision. Neuroscience is good at providing constraints on how this might happen and motivating new hypotheses, but computer vision fundamentally is useful for understanding the problem and for building and testing those ideas, and it's really a synergy of these fields that we hope to foster to actually make new progress. So this is the human brain. We love to understand how this brain solves it. Again, I mentioned we're a systems neuroscience lab. We work primarily on this brain. This is the rhesus monkey brain, not shown to scale. We love this model because we know a lot about these visual areas, as we heard from a number of talks. Particularly, we know a lot about the ventral visual stream and its involvement in object recognition. We think we know where a good form of visual representation lives for supporting object recognition, and that is in this place called infratemporal cortex, or IT cortex, and I'll spend most of today talking about IT cortex. I should remind you, this has already been mentioned, that IT sort of sits in a nice location to project those representations for decision and action around the bend into the mediotemporal lobe structures for memory. So we sort of view it as sort of the currency of cognition, these representations up here. And we also like the monkey because, unlike the human, we can study this representation and its precursor representations at what we think is the most appropriate level of abstraction, and that's the level of neuronal spiking activity. Okay, so this is all by way of background. Most of you who know me know I sort of conceptualize this problem in a way of thinking about each level of processing as a sort of a new population representation. So if you unfold this and think about these millions of neurons in each of these sheets, think of them as a new population representation. So when you see this image, it produces a pattern that looks like a photograph on your retinal ganglion cells, a nicely processed photograph, and then it's projected rapidly through the ventral stream in about 100 milliseconds to a new pattern of activity in IT cortex. A new image, new pattern in IT, so new population code. And when you were watching this movie that I showed you earlier, your IT is clicking along, following this nicely at a lag, again, of about 100 milliseconds. So again, just sort of following that along, new population patterns being generated. And what we really don't understand are the algorithms that are in operation here, and that's really the thing we deeply like to understand. So these are the tools that we use in the lab. We use behavior, we use neurophysiology. I'll tell you a lot about that. We do some computational modeling, and we also do some imaging, and I won't say anything about that at all. But I wanted to frame the two big questions that we work on in the lab. One is sort of what I call the boundary condition problem, which is defining how well can IT actually support the kind of tasks that I was showing you. What is the IT representation like, and how well can it support recognition tasks? And the other line of work is this holy grail question that I mentioned, which is exactly how is this representation computed from the retina? And you've heard many efforts to try to build up from the bottom from great talks earlier today. But that's what we view as the sort of really holy grail problem. So here's what I'm not going to talk about today. So our lab has worked a lot on the importance of unsupervised learning in building this. You heard some of those ideas computationally from Tommy earlier. And so the idea of learning about object invariances across time. We've done recent work on that that's quite exciting. I won't talk about that at all today. We've also been working on building and testing computer vision style algorithms to try to falsify them with neuronal data. And I won't talk really at all about those today either. So what I will talk about today is our more recent work on this question. So how well can this representation support recognition? So now I need to remind you of sort of what is the problem of recognition. Why is this a hard problem computationally? And the crux of the problem, if you're a climber, the crux is sort of, it's not the whole thing, it's not everything that you need to do, but it's sort of the hard part of the climb. Once you get around this part, we think the rest of it is going to be relatively straightforward. And so the crux problem as we see it is the problem of identity-preserving variation. And that is, an object like a car can present an essentially infinite number of images to your eye. And this is due to general variation, which might be due to latent parameters in the object, such as its position, its size, pose, and illumination, is sort of illustrated here, to background, potentially occluding, which we might generally call clutter, and also to interclass variation that Tommy mentioned. So there are many different types of cars. Okay, so these are all the general sources of variation, and somehow we can see through all this and sort of say every one of these is sort of has an image, has a car in it. So let me sort of give you a sense of how we like to think about this in a population level. Again, this is a population coding meeting. So here's how we conceptualize this. And this relates to things that you saw from Charles in a lower dimensional space. And the idea is if you think about any population representation, imagine the retinal ganglion cell representation here, you can conceptualize it as each of these axes being the activity of one retinal ganglion cell neuron. So here we have, say, a million-dimensional space if we think of the whole thing at once. And this is a particular image of a particular person under a particular set of latent parameters, that is pose and position and size and so forth. So that's one pattern of activity, one photograph, and it amounts to one point in this high-dimensional space. If this person undergoes a single transformation, the kind of things we need to be tolerant or invariant of, such as a transformation in pose here, it's going to sweep out a continuous set of alternative representations, or sort of alternative activity levels within this representational basis. And so if he undergoes another degree of freedom, here's another change in pose, another direction might go like this. And you can imagine, in this case, 2 degrees of freedom and the thin two-dimensional sheet of points that this particular person could give rise to under these 2 degrees of freedom. I want to point out this is a highly small part of the ambient space, a very thin part of the space, and it's highly curved, as you saw in Charles's slides. And the reason I'm pointing this out is because if the world was such that these kind of what I'm calling these object identity manifolds, here shown only in 2 dimensions, they're higher dimensional than this, but they're low dimensional relative to the space. If these low dimensional object identity manifolds, if the representation was something like this, we would call this a good representation, for instance, in the ability to separate this individual from that individual with simple sort of decoding tools, things that could be implemented by neurons, such as separating hyperplanes. And I'll talk a lot more about those later. So this is what a representation might be that would be good in this so-called visual world, as opposed to a representation. This is a simple idea where these are sort of embedded each other, and the toolbox that you have to decode it is just insufficient to read this representation, to read the latent variable of identity in this representation. And in fact, this is a simulation of a pixel basis with 3-dimensional objects of these 2 individuals. And this is meant to show you that this is only a 3-dimensional view of a high-dimensional space. But these are like 2 sheets of paper that are sort of crumpled up together. And we call them hopelessly tangled. You can't easily take a hyperplane and find a way to separate these individuals under a large degree, all the kinds of variation that I mentioned earlier. So we like to say that object manifolds, identity manifolds, are tangled. And this is due to identity-preserving image variation that I've mentioned earlier as the crux problem. So this is conceptually what's going on. We have this sort of crumpled up sheets of paper up here, if you will, of the different objects, and we have to somehow sort of untangle them and produce something more explicit up here. And again, we consider each stage a new population representation, and we think there's a more explicit representation, something that might look at the population level more like this up in IT cortex. And all the analyses that I'm going to show you for the rest of the day are essentially aimed at estimating how good this representation is with respect to solving these kind of problems. OK, so we've done, again, a lot of work recording up in these higher level areas. I won't tell you about all of that, but I'll give you some of the older highlights and then show you the newer stuff. The older highlights begin by first reminding you of what IT neurons look like. For those of you who haven't seen IT neurons fire, if you go on a lowered electrode, it's deep down here. Here's anterior IT. You have a long distance to get there to even record. You put an electrode near single neurons. Many folks, and you heard from some of them from Ed Connor's work today, record in IT in the past. And whoops, well, that's my example. What you see from IT when you do this, well, here's listening to a neuron. So this is kind of how we might collect neural data. That little clicking, that's a neuron firing in IT. We're presenting images at 5 per second. This is a bit grainy. The monkey's just fixating here. This is just to give you a sense of kind of what it sounds like in the lab when you record these neurons. So that gives you 2 senses. One in which the rate at which we present images, and I'll talk more about that later, and the notion that, it doesn't sound like much there. I hear a lot of spikes. But when you sort this all out and you randomize everything and you come out and say, let's sort out the times when we presented this particular image or this image or this image, here's what you get from this kind of site. So here you see these little tick marks are action potential. So you see this site responds reliably well, lots of spikes here, to this image, and also somewhat to this image, not so much to these images here, with a latency you can see of a little more than 100 milliseconds in this case. And different sites in IT will have different sort of preferences in this sort of image space. So you heard from Ed on the sort of the attempts to try to understand the feature spaces of places like B4 and IT, which is quite a difficult problem. We take a very different approach where we try to just understand how good the populations are for supporting tasks. So I'm going to tell you the way we do that is we first have to estimate what's the response of such neurons. So we do that by, for instance, counting spikes in a behaviorally relevant analysis window. Here, say 100 milliseconds. And I'm going to do that for all the analysis I'm going to show you later. So we just think about spike rates in a window of time. That's a time window that we can vary easily, and we do vary. But just think about 100 millisecond spike counts with a latency of 100 milliseconds. And here's the first study we did soon after I got to MIT. This was with my colleague Tommy Poggio, who's in the room here, Gabriel Kryman and Xiao Heng. And what we did was we just sort of thought up a set of objects. This wasn't sort of strongly motivated in a kind of parametric way. We just had grab bags of objects, and we had them in 8 pre-assigned categories. And we put in variation on things like position and scale, because we knew these invariance problems were things that made recognition hard. And we wanted to see how good you could do just recording from populations of IT neurons. We presented images. You saw at a rate of 5 per second to a passively fixating animal. And these are the kind of data you get out. Each of these little green boxes is sort of the mean firing rate of one particular IT site across a set of 78 images shown here, just to show you the kind of population data we get. We collect many different recording sites, just randomly sample them throughout IT. In this case, we had 350 IT recording sites. And so you have this big population pool of data. And now you can take this data and apply essentially linear decoder classifiers to it to ask how good is this population for doing things like categorization and identification. And again, the way we do that is we take an image, randomly draw sets of spikes that we would have recorded from this image from a large set of neurons, or we can vary the number of neurons. That turns it into a vector where there's a scalar element here. Again, spike counts in a 100 millisecond window. So we have an n-dimensional vector where n is the number of elements, the number of neurons in this case. That's a point in an n-dimensional space. And so we have, in this case, we have a task of saying, is this, say, a face or not a face? That's sort of a face detection task, face versus not face. And we did this, again, for 8 pre-assigned categories. I won't take you through the details. This is old published work. But the thing that was interesting, we thought was pretty interesting, is that you really only need a small number of neurons to get good real-time performance. So this is the number of sites. You get up to nearly 100% 8-way categorization performance, real-time, cross-validated, all that good stuff. And so that's kind of nice. 100 neurons is a nice number to put in your back pocket. And we also showed that your ability to do this in a way that's invariant. So if you train a classifier on one set of images, and then you test on translated or scaled versions, you get nearly the same performance, only a slight drop in performance. So very good generalization over changes in things like position and scale. And we later showed for some clutter, that's not possible with simulations of early visual cortex. So that's, again, one of the nice properties that IT has, and we can measure that with these kind of decoders. I want to point out these data were all collected in a passively viewing animal. There were no attentional cues. And these are really the first spikes out of IT, 100 millisecond latency, probably limiting the amount of feedback that could be involved in producing these spikes. So we think this places a strong constraint on the kind of models that might be at work in the ventral stream. I'd also like to say that more recently, we sort of did that phenomenology in IT, and then we sort of said, well, is it better than V4? Can we even measure that? And Nicole Rust in the lab came and measured a whole bunch of V4 neurons and IT neurons, so measured population of V4, population of IT. Again, I won't take you through the details, but she recently showed that, yes, you can clearly see readout performance differences in terms of generalizability, sort of what you'll call population invariance in IT relative to V4, and that's been recently published if you'd like to follow those details. But so that was the big picture. And this is sort of like around 2009, I'm sort of giving this talk like this. And the thing that's always bothered me is, yes, it's nice to show there's some readout performance and it's better in IT than V4. But is this sort of first spike code sort of sufficient to explain what we mean by object recognition? And, or is it just sort of, it's better than V1, but is it sufficient? Well, sufficiency implies some sort of goal or reference. And so really what we needed was to better operationally define what we mean by object recognition, measure that thing in humans, and then see how well we do. And we needed to get a whole lot more neural data. So we've been doing both of these things. And so now I can give you some preliminary answers on this question. And that's what I'll spend the rest of my time talking about. OK, so better operationally defining recognition. It turns out that on the computer vision side, we'd already been building classes of images that better screen computer vision style models on invariance problems. And the way those are motivated is if you think about a car detection task, we don't actually use real world images, unlike a lot of computer vision. We take 3D objects. We can take many of them from a class. We can render them. We can introduce more or less variations. We have full control of the world. And then we place them on naturalistic backgrounds because this really makes it difficult for computer vision systems to cheat on the background. And humans can sort of see right through this and still say this is a car. So that was a strategy we'd already been taking on the computational side. And so we used those kind of images to start to test humans and neurons. And so we assembled tasks for humans. This was an 8-way categorization task where we're going to have animals, boats, cars, chairs, faces, fruits, planes, and tables. So this is called a basic categorization task. This is just one example on one background under one pose condition and so forth. So these are just one image of a large set of images that humans have to say this is an animal and not a boat or a car or a chair or a face. We also we can vary the difficulty of the task by making it more subordinate, say distinguishing different types of cars from each other. I know these are hard for you to see at this scale. I'll show you some in a moment. And for distinguishing very, very similar faces, so a very subordinate face task, distinguishing these faces from each other. So we have a range of task difficulties with respect to shape differences, and then we can control parametrically how much variation we put into the problem. Again, that crux knob. And so we vary both of those things, and we end up with essentially 29 kind of benchmark tasks. So we're going to get 29 d-prime numbers out of humans in distinguishing, say, this is an animal versus not an animal, or this is this face versus not these faces. Those are the kind of d-primes that we collect. And I'd be happy to talk about those details in question time. To give you a feel for that, I want to remind you that we're doing this in sort of what I call unfettered viewing conditions. We're showing images for 100 milliseconds. There's no mask after this, so we think it's very comparable to what you might get if you were saccading, taking a sample of a saccadic interval, as I mentioned earlier. For the image span 8 degrees, just so people know. Again, we're working on about the central 10 degrees of vision. And here's an example of a few images. If you want to get ready, they're going to show up over here. OK, anybody see what that was? Car. OK, that's a hard one. Plane, right? OK, I didn't even tell you what you might see. Face, car. OK, so it's a little weird. There are cars floating in the air, but you mostly have no problem doing this, right? And you can see the variation going on there. And we don't think this is the perfect measure of object recognition, but it's better than no measure. So we had nothing really as a basis before in the field, and there's various sociological arguments for why that's true. And I could talk about those as question time. But really what we have now is 39 human subjects. We've tested with 23,000 images on tasks such as these. And that's given us a set of psychophysical data in these tasks that essentially define what we thought was a desperately needed operational bar to at least say, how does IT do? How are computer vision models doing relative to that? Where do we stand in the field of solving object recognition? OK, so again, I've told you how we also need to get a whole lot of neuronal data. I'm going to go through this quickly in the interest of time. We were involved in a Dartmouth collaboration with Jack Galan, also here in the room, to collect data. And so we developed methods to put 96-channel arrays and surgical methods to get them down in the ventral stream. So we're implanting sort of triple 96-channel arrays. We can do them in V4 or along IT, usually just 3 at a time. What's nice is we can record lots of sites simultaneously, but we can get lots and lots of data over many, many days. And this is sort of our data rate in terms of number of images shown per site per year. And this is sort of using old methods that we'd have to sample serially. And this is sort of the sort of acceleration that we got out of using these kind of arrays in terms of data volume. So here's an example of the kind of data you get out of these things. All the data I'm going to show you about today is multi-unit data. Again, I'd be happy to talk about that in discussion. These are multi-unit responses in response to, these are 8 images of a set of, say, 2,500. There are about 10 reps in there as different rows. So these little dots you can barely see are spikes. And you see these are images that happen to contain an animal. I'm just showing you this to tell you about how we get the data. Again, passively viewing primate, no attentional cues, 100 millisecond duration, 5 images per second, all the same as before, 10 to 50 repetitions per image, and we just randomize all these images when we show them. And then we count spikes in a window. In this case, it was 70 to 170. So again, a 100 millisecond window at a latency of about 70 milliseconds. Okay, and that gives you this kind of vector. Again, these are the mean responses for these particular 8 images, mean over these repetitions. Remember, we had other categories, and this is just some of our data. I'm just trying to give you a feel for the data. And we didn't just have 8 images per category. In fact, we had something like 640 unique images here in this particular set. Some of our data sets have 2,500 or more images in them. So this is a feel for the kind of data that we get. And remember, this is just one site I'm showing you. This is a site that sits among 128 sites that we're collecting simultaneously along the ventral stream, again, with those chronically implanted arrays. And so these kind of data allow us to start to estimate how good is IT doing or V4 doing relative to those human data that I mentioned earlier. So now in the whole rest of my talk, I'm going to take you through, this is all new results, and I'm going to tell you kind of how we play this game here. So the logic goes like this. So we have these 29 tasks. We've done all these images, tested a bunch of humans. So the values on this axis, the performance of the human subject, these D primes, these 29 D primes, they're not going to change. They range from pretty low to pretty high. 6 is a pretty high D prime for those of you who know psychophysics. So we have this range of abilities on these tasks. And then we're going to bring in various neuronal data, computative codes of how you might read that data to support this task. So we'll get a D prime. So we have commensurate measures here. So how good is this code doing on each task? That's going to give us 29 points on this scatter. So we'll take in IT data. We might try some V4 data. We'll take some computer vision feature sets, and we can keep playing this game and see what kind of codes work and which don't work. And here's kind of what work would look like to us. So here's 29 points. This is just kind of just conceptually. These aren't real data. So work would look like this, where you'd say that we might call this the true code. It has equal performance as the humans on this dashed line. And when the human's very good at a task, say one of these tasks here, the code is very good. And when the human's very bad, the code is very bad. That's sort of what the ideal code might look like if you think it really is the population code supporting this human performance. So that's the true code. Let me show you what some other codes might look like before I show you the data. So here might be a poor code. These are pretty easy to generate. So again, human subject values don't change. So everything's just moved down the axis here. So this is performance of a putative code where it's basically terrible. It has very low D primes, and it has not much relationship to what the human's doing. Everything's mostly below the diagonal. Here's another code where you say, wow, this code kicks butt. Huge D primes on all tasks. In some cases, it really even beats the human on tasks. So it's well above the diagonal. It doesn't have a consistent relationship with the human. We don't have a name for this code. I call it the DARPA happy code, because this is sort of the implicit goal of computer vision or DARPA that says, we just need it to perform well. And at the end of the day, I'm not sure how much they care about whether it matches the human performance. Then there's another code which might look like this. It's highly covariant with the human, but it's actually not so good, right? So DARPA wouldn't like this, but I call this the neuroscientist happy code. So implicitly, neuroscientists have been doing this for some time, trying to look for neuronal correlates of behavior, sort of correlates in this sense. And there are many beautiful examples of this, especially in subjective magnitude domains. And my favorite example is Ed Connor's beautiful roughness work in the somatosensory system that sort of goes after this kind of approach. And so, this is just to remind you that we're thinking about both of these goals of what your code should do at once. So really, a true human-like code must succeed in at least two ways. It must have good performance in this DARPA sense, and that was the sense and sort of way I brought you to the problem. We'd like it to get to human-level performance. We also think it should have what we call consistency of the code across all the tasks that I showed you. And that's more in this neuroscientist set. So it's just so the human pattern of goodness and badness in performance. And so the true code in a plot like this, if we could index that, should live up here somewhere. And so now let me start to remind you. Here's the true code would be up there. We can now start to put numbers on this. So this is going to be what we'll call the mean Turing ratio. So take each task d prime of the code over d prime of the humans. Take the mean across all of this, or the geometric mean in our case. And this is going to be just a simple Spearman correlation to just sort of measure the correlation along here. And you can see in this case, this is going to give you a point up near this corner. In this DARPA happy case, it's going to be up here. And the poor code is down in this corner. And neuroscientist happy code is over here. OK, so now I'll show you the data. OK, so now that I've set up, what codes are we testing? Well, I already mentioned the code bases that we want to talk about. IT data that I mentioned, we collected V4 data the same way, pixels as a reference, and computer vision style things, sort of V1-like simulations, and a bunch of computer vision algorithms that we implement in the lab just as references. And so, and then among these codes, there's still many details. How many elements or dimensions are you going to include in the code? So that's something we're going to vary. And here's some of the ranges that we've used here or projected here. How you read the code, even though I said linear classifier, we're going to use an SVM-based classifier. How much training data are we going to give it? So there's sort of many potential codes hidden in here. I'd be happy to talk about that. There's sort of more simple codes like just find the best element or the 90th percentile best element or the median element or other sort of, let's say, like linear classifiers, but perhaps learned in a simpler way than you would learn the SVM. So these are all codes that we don't know. Are any of them good? Are all of them good? Are they sufficient to explain human recognition performance now that we have that data in hand? So that's the space of codes. I'm going to show you data from these two codes to start, and then I'll show you the results from all these codes. So here's the game again. Remember, we're going to bring code in here. So here comes the pixel code, all nice and robust, saying I'm going to kick butt. Pixel code, image pixels, SVM in this case, 20K pixels. We select those so that there's nothing dumb about the pixels not responding to the images or things like that. And so here's what actually happens. Here are the pixel performance, the d primes of the pixels versus the human. And what you can see is there's a lot of tasks, as you might expect, where the pixel's very bad at the tasks. So it looks like that poor code I mentioned. The reason it's bad is because these tasks have a lot of invariance in them, which is really hard to do on the pixel space, which is that tangling problem I mentioned earlier. And as I mentioned, we have tasks where we put low amounts of variation in some of these tasks. So for instance, here, these are 8-way face tasks where the faces are barely having any variation at all, essentially 0 variation. And the pixel code kicks the human's butt, right? An SVM on a pixel space, no problem. This is like barcode reading in your sort of grocery store. Computers are good at pattern recognition when defined in this way with low variation. This is useful for us. It might make DARPA happy in some regimes, but it's useful for us because it shows us the importance of consistency, that you really want a code to actually fit all of these data. And the pixel code has revealed itself to almost be too good in some regimes. And so this gives you essentially, when you quantify this, a pretty low mean Turing ratio in terms of DARPA goals, 0.16 because these help it and these hurt it. And the consistency is pretty terrible at 0.29. So the correlation, the Spearman correlation is only 0.29. OK, so that code slinks off and didn't do so well. And then I'll bring in another code. So this is sort of the other extreme. This is an IT code, SVM. This is 58 elements that we collected from IT with these arrays. And here are these data here. So what you should see from this is, first of all, we were quite excited when we saw this because, first of all, we're not that far below this diagonal. In some cases, we're above. So this would be sort of matching human performance. This is just 58 neurons that we happen to collect. You can see also quite good consistency across these tasks. And so you can quantify this, again, a mean Turing ratio. It's about 2/3 what humans will do with this particular IT code, and a consistency of 0.8, which is quite high. So now let me show you those two codes on this space. So here's performance. Here's the sort of DARPA or Turing ratio goal. And here's a consistency goal. Pixel code, pretty bad in this corner. Poor code. IT code sort of up here, not quite in here, but approaching that. So let me take you through all the codes that we've tested so far. All the pixel-based codes live down in this corner, as you might expect. They're quite poor. V1-like codes, these are simulated V1. They sort of move things to the right. So better consistency, slightly better performance. V4-based codes, even better consistency, and now some of these codes are reaching up to high performance, and now here are IT-based codes. This is a series of IT-based codes, and you see further to the right, and some of these codes reaching quite high in performance. So you can see things get better along the ventral stream. That's interesting. There's some codes that might still be in play here. So this is sort of the lay of the land, and we now have a framework to start to sort of analyze these things in this systematic manner. And I'll also say as an aside, if those of you who saw Ha's poster, we'll bring up computer vision codes and where they currently sit. This is another good reason to have these kind of plots, as we can sort of say how good are we doing on the computer vision side. They're down here in sort of the mid V4 range or slightly better than V1 for the moment. Okay, and these aren't necessarily the latest, greatest state-of-the-art models. These are just models that are published in the literature. Okay, so let me sort of say how we interpret this data at the moment. So there's some good news from these data. Sorry, I'm stuck. So I take this as good news right here. Our data, as you take it, now we could quibble with this, but our data essentially say all of these codes are wrong. None of these codes support human recognition. Therefore, they are not representations of human recognition. That is the falsification model of how to do science. Say, these codes are not right. Okay? Now, you might say, well, there is some computer vision code that is right, and that is probably true, but these particular computer vision codes are not right. And some of these IT codes are not right, and some of these V4 codes are not right. However, there's more good news. There are some codes that look like they may be right. So there's some good news from the coding standpoint. There's at least one code on this plot that's still standing. This happens to be an IT-weighted sum with an SVM, in this case with approximately 500 neurons in it. And I'll say more about that code in a minute. So I just wanted to give you the lay of land of where we sit in terms of how well we can explain object recognition in the ventral stream. Here are these codes that I sort of started with, and here's kind of where we think we are. Most of these codes have been falsified. This one here is still standing. Some of these we're not ready to commit as whether it's standing or not. And I should say all of this is still preliminary. Even though I'm putting this X's here, we're still collecting data from new animals, but I don't expect this picture to change much. In fact, going forward, what we're doing now is we want to do things like uncertainty due to our neuronal sampling, exploring codes. This is sort of SVM codes. Let's explore codes that have what I'll call more biologically plausible reads, perhaps still linear classifiers, but perhaps under sort of a more bioplausible conditions. And also explore other computer vision codes that we and others build. And so let me sort of also point out that this framework for testing codes that I've sort of presented, we want to emphasize this idea of consistency, which was essentially that neuroscientist-motivated ideas being important to sort of supplement what's going on in this sort of more computer vision style or DARPA style. And the reason I want to point that out is that if you look at these particular two codes here, what they are basically the same kind of code basis, but increasing the number of code elements. So it's pretty easy to take codes that are kind of lower and increase the dimensionality and the amount of data that you train them with and move them along this performance axis. But it's harder to move them in this consistency axis. At least that's empirically what we've seen so far. And in all of these kind of codes, it's very hard to move them this way. And we can also, here's another example of that up here in IT, increasing from this code to this code by just increasing the number of code elements. And so here is that actually shown in a graphical format. So now this is Turing ratio, which is like performance on that the same y-axis that was on the last slide. And now this is the number of code elements on a log scale. So here's one IT code with a particular readout method. And you can see that this is the code I actually showed you about. So this is an extrapolated IT code based on the data we had in hand, right? So we were estimating a code of about 500 IT neurons to reach a 1.0 human level performance under this extrapolation. V4 codes would take nearly 0.1 million neurons, and almost all computer vision algorithms would take many millions of neurons at the moment under current implementations. And this is one that we built in the lab, and I don't mind beating up on this thing. We know it's not the right model to explain things. I should say there's roughly 10 million sort of projecting neurons out of anterior and central IT, so that's a sort of number to have in your head. But this is a kind of number that you could imagine single downstream neurons computing, so we like that, but this is the lay of the land of where we sit right now. So the big picture that you should have in your head from what I've presented today is that there's this problem of tangled information. We think it's explicit and untangled. I've shown you phenomenological evidence that we found a long time ago to suggest that, and now I'm sort of beefing that up with showing how explicit it is with respect to human performance, and that requires an exploration of the potential code readout mechanisms, and I've shown you the beginnings of that, and I think we're really only at the beginnings of exploring those potential codes in this falsification framework. And I want to remind you that everything I showed you today these are easy to read. These are sort of linear readout codes, nothing fancy going on there. These are first spikes out of IT. I showed you those time windows, and our previous results show that you can get by with even smaller time windows, so we can play with that going forward. This is all passively viewing monkeys. Again, taking a lot of sort of attentional arguments out, perhaps and the fact that they're first spikes, minimizing the amount of feedback loops that you have to use to get this representation out. So we're not claiming that feedback isn't important. We're not claiming that attention is not important. All we're claiming is parsimony says, here's how far you can get with respect to humans out of IT. And that's what our data can sort of constrain and frame the problem as a possibility. So some codes, thank you, some codes are sufficient to explain human performance. That's what I've spent most of today trying to tell you, is that a lot of codes can be falsified, but there are some that still seem reasonable to us that are sufficient in IT to explain even human-level performance from these monkey IT data. For the aficionados in the room, and this relates to what Charles said, I've only been talking about sort of measuring shape variables out of IT, sort of putting hyperplanes in here to say, is this face and not this face, for instance, in this drawing. But of course, there are other latent variables that we think are still coded, and that's why I want to emphasize that we don't think IT is sort of throwing away things like position and scale. In theory, you could decode these. We've showed this with Tommy in an earlier study, that you still have this kind of information in IT. So don't think of IT as sort of only being sort of grandmother cells for identity. It's essentially factorize these latent variables in the language that you heard from Charles. That's what we think this code is like up here. I've just been talking about how good it is on this shape or identity axis in terms of these recognition tasks. So I think this is my last slide. I'll just give you my conclusions of what I've tried to tell you today. So invariance is the crux problem of object recognition, we think. I've given you sort of a conceptual way to think about that as the untangling of object identity manifolds. And thinking about it that way is especially helpful when you start thinking about the unsupervised learning rules that you use to sort of unwrap those object manifolds. The ventral stream we think untangles rapidly, and we know this sort of at what I'll call a phenomenological level. We can go up to IT, measure it, use a measure of separability of these object identity manifolds. And again, first spikes out of IT, even without attentional queuing. Some IT population codes, as I showed you today, this is all sort of still preliminary, but some IT codes seem nearly sufficient to explain unfettered human performance. And again, all V4 codes we've tested so far and all man-made codes tested so far are insufficient to explain human performance. So that's a nice contrast. I also want to remind you that this measure of consistency, this neuroscientist-motivated measure, brings power. You don't want to use it on its own, but if consistency in combination with performance brings new power to the ability to sort of tease out which codes are true codes and which codes are perhaps not the correct codes, as I mentioned in the last few slides. So let me just end by thanking the folks that did the work that I presented today, especially Najib Majaj, the postdoc who basically has spearheaded this entire project, and Ha Hung, who's here today and has been doing a lot of the computational analysis that supports it. And he had a poster of those. You saw it today or can go back and see it, especially comparing those codes to computer vision codes more precisely. This is Ethan Solomon. He's an undergrad who actually collected a lot of that psychophysical data that I described. And this is Nicholas Pinto, who implemented a lot of those computer vision models, which is no small feat, to test them on the exact same images that we were running on the humans and on the monkey physiology. And I'd also like to thank Tommy for sort of motivating me along these lines all the way through. And thank everybody else in the lab and my funding agencies. And thank you for your time.
Questions? Ed?
So, Jim, can you explain more about the codes that were falsified and maybe give an intuitive idea of why they fail?
Yeah, the easiest way to do that is I can go to the pixels one. So let's look.
I was thinking of IT codes that get falsified.
Well, so those are often, here, let me put this up. Those are mostly, I mean, Ha could chime in here as well, but those are mostly falsified for reasons of performance failure, right? So if I took 2 IT neurons, it's going to have pretty low performance. So I'm going to falsify a code that says, I'm going to take 2 IT neurons and explain this human level performance, right?
So the difference is
I'm not falsifying IT. I'm falsifying this particular read of IT. So
the spread on IT models is mainly a matter of number of units?
Well, that's an important free parameter, right?
Sure,
we keep it as a free parameter, and our constraints are like, well, how many IT neurons are there total? That's sort of an upper bound. How many, if I'm a downstream neuron, how many could I read at once, right? Those are sort of the ways you might consider constraining.
Sure, yeah, I understand. But when you talk about falsifying codes, I assume there's something else besides just number of units that you're falsifying.
Well, so if I show you the particular code instances, let's go to the lay of the land of the code space. We have to sort of define what we mean by the word code in this sense, but each of these dots I'm calling a code. And so I say the code is based on IT or V4 or V1. That's the basis set, but that's not the code. That's just the basis. And so let me sort of see if I can get to. OK, so here's a space of codes. And you can imagine expanding this as big as you want. But you've got some IT basis. Now, you have to assume we've sampled this reasonably. I mentioned that as some direction going forward. You can vary the number of elements. Here's 2 particular codes we just happen to run. But of course, you could sample everything. And I showed you the extrapolation. You can vary the way you read the code within constraints. If you start putting super nonlinear classifiers on it, you've sort of lost traction at all. Right? And I'm actually a big fan of dropping strong classifiers that give you big performance, but sticking with these sort of simpler kind of classifiers as a sort of as a better way to get started, to see what's the simplest classifier you can get by with. So our answers are going to look something like this simple classifier with 1,000 neurons could support it, or this complex classifier with a few less neurons could support it. And that's kind of the space at which this data will at least constrain the problem for the moment. We're going to have to bring other things to bear to go further than that. We're going to sort of show you the manifold of solutions, right?
Right. So then the codes that you actually consider falsified, right? Yeah, there you go. So best element is on the border. Best element is...
This would be like, take these 58 elements, find the one that has the highest D prime on cross-validated data, and test its performance on new data. So it'd be like, imagine you're looking at IT and like, my job is I'm prefrontal cortex, and I sort of somehow sample the neurons. Again, these aren't really bioplausible in that sense. And say, oh, here's the best one. Right now, HA does that in a computer. But you sort of imagine that somehow you've got to read the IT and say, oh, that 58 random samples, I get to sample 58. I choose the best one, and that's how I do my detection of cars.
So choose the best one for all time, or the best one for
best one for each task. So every time I build a D prime, I say, I'm a car detector, right? Or my job. So each of those 29 things is to build a single detector for each
single just
label.
One site.
You conceptualize it as a simple downstream grandmother cell if you like. I don't think it's going to be that quite simple.
So that's actually close to being a check, because it's still got a question mark on it.
This one, no. The reason I put that question mark is because you have to know how the best element's going to. Best element is sort of dependent on the sample size strongly, right? So
right.
If maybe I'm missing key elements in the sampling here, right? So. But you could also start to say the limit, who's ever reading this code could never really find the final best element in IT, but it might do something like screen the elements and find the 90th best one. Again, that's sort of getting into the space of what are the plausible ways in which this thing could actually be read by a decoder, which I think is really what we need to do with these data.
So just to make this explicit, or more explicit for me, the code, the basis functions of the code for the IT neural data and the V4 neural data were just the neurons.
Were the actual neural data.
And the codes only vary along 2 dimensions in this experiment, which is the number of neurons you're allowed to integrate over and which neurons you select.
There's a 3rd dimension, which is how much training data you give. So there's those 3 sort of free variables right now, right? And we think the most interesting one is a sort of like, well, the combination of how much training data you give and what kind of learner you're using. That's the space that I mentioned. We like that to try to be more bioplausible would be the goal. Right? Not just say it's a linear classifier, which is bioplausible, but learning like an SVM with gazillion images is perhaps not bioplausible. Right? So.
I was just wondering, have you tried fusing the codes and seeing if the fusion works?
Not yet. So what would you suggest we do? Just
the different levels that you have.
You mean so now you're building codes on top of codes? Sort of machine learning style. Yeah, I mean, that seems to be the tendency of machine learning, like, oh, let's get more juice out of it. I kind of push back against that, right? Because the more you need to do that, the less you're going to argue that the code lives in IT, right? It's like you have to keep the toolbox simple, otherwise it isn't the code, right? Because then I could say, well, the code lives in the retina, right? Just stack a ventral stream on top of the retina. The code's right there. So code has to be framed with respect to like the sort of toolbox that you're allowed to bring, right?
Cornea.
Sorry?
It's in the cornea.
Yeah, right. It's sort of out in the world.
Can you use the microphone, please?
The number of the number of neurons you sample, it's circumstantial in the sense that it depends on you. You might actually find by accident the best neuron, then the number of neurons would be without any consequence.
Well, that's kind of why I was sort of mentioning, you're talking about uncertainty due to neuronal sampling. So we can do that by subsampling from the pools we have, but the bigger our pool gets, the more confidence we'll have.
That's right.
I want to put error bars with respect to that uncertainty on these kind of answers, and that's what we're working on now. And we're not going to be able to do that in a very strong form because you always hypostulate, hey, there's dark neurons that your electrode can't see. We could always sort of make stories like that, right? So there's some limits to that. But within coverage of IT and large numbers, I think we'll be able to squeeze that pretty tight.
So, in other words, it doesn't follow that it's a population code.
Well, you can't prove that anything is, we can't falsify the idea that there's some best neuron out there somewhere that is what's actually happening, right? All we're trying to say is what is the space of things that could be happening, check, and the space of things that cannot be happening, X, right? And that's a sort of falsification model of science, really, right? And that's kind of what's implicitly under the hood. And again, I present Ed's example of coding of roughness in the somatosensory system as sort of a good example of that for me.
Jeremy?
So you've reduced everything, both behavior and neural codes, to D prime, which is nice because it's straightforward. But I'm wondering, maybe you have already looked at, presumably you get a lot more data, more readout information from both the codes and from the human behavior, things like false alarms and confusions and other kinds of things that could provide additional forms of falsification. And maybe once you include enough things, everything would fail. But that'd be an interesting if you guys
right.
looked at
It's sort of a question of, for instance, we're doing this per task. We could start to go per image ultimately, right?
Sure,
it's like full
sure.
confusion matrix, right? We're collapsing along certain ways. In fact, we took an 8-way task it was actually run as an 8-way task, gives us 8 D primes, right? That's sort of like 8 tasks, but it was actually 1 task when the humans ran it. But yeah, I mean, that's the way forward. That'll be now how strong is your psychophysical data, right? But that's the consistency direction more and more refined, right? And I think that's sort of along the lines of what you guys are doing with metamers, and I think that's kind of what should happen, right?
There's a question back here.
So, I think this is on. Yeah. In the beginning of your talk, you mentioned you recorded from like 350 cells or something like that, but then for your model, you're using 58 cells. What's special about those 58 neurons?
So those 350 were sampled serially with a much smaller image set. And that was sort of using those old techniques where you can go and serially sample that. That's a study we did with Tommy 5 years ago and sort of paints some phenomenology. But now we needed much bigger image sets. And so we have chronic arrays. And, they're not all the electrodes give you good data, right? So now you're into sort of methods details. But that's why that 58 number is there. Of course, now we have another monkey cooking right now in the lab. So we can keep making that number higher. But so one advantage of those arrays is they don't give you huge volumes of samples to the sampling question. But you can, because they're chronic and relatively stable, you can get very large volumes of images and essentially tasks out of them, right? So that's sort of how we've been leveraging them. Not the way you would think, which is let's do some fancy correlation analysis because they're all simultaneous. We could do that. We don't think that's where the fun part is. We think the fun part is sort of pushing the kind of a volume that you can show to that population. So that's what we've been doing. But it's still quite small. I agree. 58 is tiny.
No, that's not my argument.
No.
I was just wondering if you were
Why
maybe only sampling cells that had a firing rate above a certain threshold, and I would argue that
No.
the
But one thing that I should mention with regard to your question is we showed to these same neurons things that we showed to old neural pools that we have using other methods, and then we're trying to sort of make sure that we get the same decodes per method, and that gets into the single unit versus multi-unit question, and that's sort of trying to make a bridge between these methods and old methods. So far, that all looks quite good, but there's still work to be done there.
There's another question back here.
I have a quick question about the data set. In the data set, you have the images of the objects sort of placed in random backgrounds, right? But that sort of gives an advantage to neuronal circuits because you give it a kind of segmentation already. Would you consider, running the experiment with just the foregrounds, that is just the objects of interest, without placing them in any background?
But you're saying this gives an advantage to the neurons? Relative to the machines, or what is your...
Relative to the computer vision.
But that's kind of our implicit logic, right? It's like, let's not test the humans in a domain where we know where to do that, where they stink relative to machines. I showed you sort of some of that with the sort of faces don't do variation. I don't need to help the engineers with that task. I need to go to where the humans are sort of show their prowess, and that's sort of the argument there. If you say, well, that's not fair to the machines, I'd say, well, then the machines are lacking, right? I mean, you're right, but so that's my logic why we did that. It's not really unfair in the sense. But if Hadi, you guys, I think we've shown some of these without backgrounds. We have some data like that. In general, everything does better, right? The machine systems do better. So I don't think we've analyzed it in any systematic way. We have some of that data. In fact, the original data I showed you that I did the work I did with Tommy, those were no backgrounds, right? In that sense, we always looked at it and said, this isn't interesting from a computer vision, sort of strong computer vision point of view. It's interested from a conceptual point of view. There is variability, but just showing it quantitatively of how well you can do, where machines are actually having trouble. That was sort of what motivated us to do this, is to connect to that community. And computer vision folks will tell you, give me no background. We can solve a lot of these problems with no background with a sort of V1-like model, right? So we need to kind of put those foils out there. And I think that's sort of the power of what we're doing, is that we kind of understand where things are hard from that computer vision perspective, and that's sort of informing where we should really seek answers from the primate brain. At least that's what I hope happens.
Could you comment a little bit on your feeling of what the true model is going to be like in computer vision? There are these sort of feed-forward models followed by classification, but there's also generative models with iterative, recurrent fitting. So the ones that you tried, so which category do they fall in, and what's your feeling as to what the correct model will be like?
Well, so there's this funny, that's the nice dichotomy to paint, but like almost any model, even a quote feed-forward model, has feedback, because you put in a normalization circuit. Well, how you do that? There's some local feedback, right? So we have to be careful about our words. But I think if you took a strong form of feedback, I make eye movements around the scene, and I sort of took that off the table right from the beginning. I had no overt feedback. That's not the domain of the problem that we've defined. But I think the really interesting space is in that 200 or 100 millisecond window, how much sort of cortical to cortical sort of talking is going on that really is sort of effectively reflected. Even though I say first spikes out of IT, some of that could still live there, sort of in that time scale, sort of tens of milliseconds of sort of going back and forth. And I'm sure there's going to be some of that's some degree of importance. I don't know how important, and to me the way to do that is for people to actually build models. Tommy does, and you heard from Charles and Arrow's group, is sort of you build these models, and then you see what works. I mean, I don't know how else to sort of, that's sort of the way we can start right now. From the experimental side, we're working on tools to try to inhibit neurons with fine time slices, like optogenetics allow you to do this potentially. That's still, sort of going to be some time, but you could imagine teasing out those circuits a bit more that way, the importance of sort of that time scale of feedback. But to my guess is like, I'd rather not guess. I'd rather just say I like parsimony or Occam's razor and say let's assume it's feed forward until someone proves it's not, right? So that's just a religion of parsimony, as opposed to a religion of beautiful generative models, right? Which are both just religions at the end of the day. So that's my guess, right? I like the feed forward class models until they break, and of course they will break. If I ask you where's Waldo, you're not going to do that with a feed-forward network. Or is this a picture of my mother drinking tea under a sofa, under a tree in the park? You're not going to do that with a feed-forward model, right? So it sort of depends on the task, really, of what we mean by this. And I think, Tommy's group has sort of pushed the class of models and see how far they get. And I sort of agree with that sort of strategy, just to see what happens, how far you can go. And it's not, and that's, I don't know, if I answered your question, Nico, was that?
Arrow?
If we put on our neuroscientist hats and we put the emphasis on the consistency axis and ignore the other one, since the other one depends in kind of trivial ways on things like the number of elements, you haven't really falsified that the computer vision algorithms can't be consistent. So what if you build into the objective function something that really tries to choose weightings that match the human behavior?
Right. So here is the crux word is you haven't falsified the algorithm. To me, an algorithm is you got to put the piece of code or the box on the table, implemented, ready to have an image and get a response, right? Otherwise, it's like a class of models. It's an idea. It's not action.
I'm with you, but wait. I'm trying to make a more specific suggestion. So you choose weights, let's say in the SVM, that are optimized for performance, correct?
That's part, again, I wanted to kind of get rid of that part, but you're right. In theory, there's a sort of like this thing sitting on top.
But that's what I'm getting at.
Yeah,
right?
okay.
The objective function is performance, and yet then you're comparing them on two axes, performance and consistency. So you don't do anything to try to make the algorithm
optimize
consistent.
the consistency.
And so you haven't really proven to us that if you had tried to make them consistent, that you couldn't. And I'm wondering if that's plausible.
Yeah, so maybe that's a fair point. I mean, maybe the better strategy is like, it's ultimately a guess of like, at least the performance feels like I can motivate that from an evolutionary goal standpoint, but you don't think that who's ever up there, whoever the queen is or reading these things, isn't going to, say, I need it to be consistent with what? With some human data around this.
But the brain has to satisfy other constraints that you're not including.
But where you're going is that's exactly where I think it's interesting. We start putting ideas about how is learning, what kind of learning should happen, and what's biologically plausible, and throw those on the data, and then maybe consistency emerges for free, right? That would be the sort of take these principles, compete them against each other, and I would still call those principles with neurons as sort of a code, right? And I haven't falsified, sparseness or all kinds of principles we might throw at it, but we have to get specific implementations of those, and I think that's what's exciting going forward, exactly that direction. That's what I tried to say, maybe not in a clear way, but I'm sure I could use help from some of you on to how to do that in the best way.
Quick question. So you have this argument against complexity in terms that, let's use the simplest models, but let's say even that SVM itself is quite complex, especially for multi-class. It's not just separating boundary, right? How, depending, either it was pairwise classifications or one against all.
These were all one versus rest D primes, right? So we don't do the multi-class thing.
So you do all the pairwise.
All the one versus rest, right? They're
Okay.
all single one versus rest D primes.
Right.
So that's good comparison to the human.
Thanks for the answer.
Okay.
But additionally, would you consider more complex models in terms of nonlinearity, which are actually plausible? Let's say all the RBF functions, which are pretty much distance between template and whatever you measured, right?
I think
which are simple.
I sort of answered that question. You're going to let the horse out of the barn or whatever when you go there, right? Because then we'll just say the code is on the retina, and we just have to sit around and think of the best machine learning nonlinearity, and we'll call it the ventral stream, right?
But it wouldn't generalize. I am not talking about really complicated.
If it was a ventral stream, it would generalize just as well as a ventral stream, right? So you pushed all the work. That's why I'm pushing back against. I don't want the SVM code. I want some simple decode, right? That's what a good code means. These neurons, not these neurons projecting through a crazy nonlinearity. It means these neurons explain that. That's what neural code.
I'm talking about as simple nonlinearity as k-nearest neighbor.
Okay,
so
that's a particular type of, that's a reasonably biologically plausible simple readout rule.
Right.
So there's some classes, so sorry, forgive me, that's a kind of nonlinear which I think could be allowed in without fully opening the barn door, right? We have to sort of navigate that carefully, but that's an example of one that I think would be okay.
Okay, I just don't want you to stop with really, really simple. By exploration, you might get actually...
Well, SVM is not simple. I think that's...
Well, the linear is. I mean,
the linear part is simple, but the learning part's not simple, right? And the images, as I mentioned, was a third parameter floating around. Well, both of them are important, right?
But why didn't you use a linear discriminant analysis or official linear discriminant and just do the projection? Sorry. I mean, it should do just as well as the linear SVM.
Versus what? What's the
linear discriminant analysis or official linear?
Yeah, that's what I'm saying. Like simpler linear discriminants.
But that would be nice because you're projecting
in the past when we've done those
product.
analyses, they usually don't change the results much.
Right.
So,
but then you can get away from the SVM and the.
To be honest with you, the reason I did the SVM here is like I wanted to show DARPA. Look, there's a number up here that can be high, right? So
it's DARPA
sort of like
happy.
Yeah. So they can motivate. Say, if you could build this feature set, you would, in theory, be able to do these kind of tasks, right? We don't know how to build that. It's the holy grail. But so there's sort of, I would send Ha to say, drive that number as hard as you can without being unbiased, right? So we do maximal training with the best learner that we have just to pull that number up that axis, right?
But if you're sticking with the linear SVM, using Fisher linear discriminant would have given you almost exactly the same performance.
Yeah. Okay. So
I'm just saying you don't have to worry about all the complexity of the SVM and the optimization and the margins.
Okay. So we should talk more offline.
The SVM is very simple. I mean, it's online learning with a little bit of regularization. That's it.
But I don't disagree. But he's talking about wanting to simplify it even more. And I'm saying if you're going to have a linear classifier, just use a Fisher linear discriminant, and then you don't have to worry. But he seems to have a problem.
No, I don't have a problem. I'm just no. I would like someone to draw me as a neuron and say, "Here's Tommy could do this better. Like here's a neuron. Here's how the regularization is going to work in the downstream, and here's how it's actually implemented in a piece of neural hardware, and it's sort of in that class. And like which supervised learner is most biologically plausible, and have a debate about that. And I'll be happy to use whatever you guys say, and I'll report a number with that learner.
Right. But in fairness, the Fisher linear discriminant is an inner product, and the SVM you have to worry about margins and things that are a little bit more.
Margin is an invention of marketing invention.
No. You want to use the Fisher one because it has a direct interpretation in terms of D prime. There's a good reason for doing it, but I agree because these data probably have a lot of noise. Probably it's not going to make any difference in terms of the results. So, but I think Jim's main point, which I completely agree with, is you are only allowed to do linear decoding, because the minute you do some decoding that's nonlinear, then all bets are off. And the only thing you're ever going to have any hope of interpreting is a linear decoder. So you can have all classes of linear decoders that you want, but it has to be linear in the parameter space.
Isn't that a little dogmatic? How about a nearest neighbor?
Well, that's what I think the previous question was.
Yeah, what Yarek suggests. Why isn't a
It
nearest
just has to be
neighbor,
linear. As long as it's linear in the parameter space, you're okay.
Well,
as long as it's linear in the base, linear in the basis functions.
Well, nearest neighbor isn't, right? So, nearest neighbor doesn't meet that criteria.
It's just
so right. So that's why I'm saying, well, that's sort of, but it's potentially.
Well, it can, because you can basically redesign the model. What you need is then to propose a model that has an n. You know, it's not just an NL, it's an NLN, but then you apply a linear decoder to it. So, my point is, if you want to do nearest neighbor, you put it in the encoding model and then apply a linear decoder. It's just, if you start allowing nonlinear decoders and nonlinear encoders, then you end up just confusing yourself.
Okay, on that note, are there any questions?
Yeah, I had a question. And a small note before the question. Small note is that if you want to do Gaussian support vector machines, which is, from the approximation point of view, quite powerful, it's just normalized dot product and then a linear online learning. So it's very simple to do. Anyway, that's a comment. The other one is a question. It's we're looking at a poster also with Hung, and there are quite a few rotations.
You mean in-plane rotation or out-of-plane?
No, in on-plane rotation.
In-plane rotation, yeah.
Which none of the computer vision models have been built to be invariant to, but mostly not because it's not possible, but for, and related to what I was saying this morning, that would be something easy to do and to try. And I imagine performance will increase. Do you have a feeling how much? Is it
I don't know. So that's one of the things I think we would like to do going forward. Is like the advantage of these having these latent parameters is we can sort of tease out. Okay, let's take the images and design just an in-plane rotation task, or sort of mix and match different types of variation, which are right now they're all bundled up together, as you sort of saw.
Yeah, no, but I mean, bundled up is fine. What I mean is the computer vision algorithm that you have.
Try to build in rotation.
Not try the rotation, but simply because.
Yeah, no, that's
laziness,
actually
no basic other consideration.
Yeah, so when we look at our sort of current little V1-like models, we say where it really is getting hurt is on the rotation, right, as you say. Yeah. So maybe that helps paint a way forward to improved computer vision models, and you guys can probably do that better than anyone. And so that's I was thinking you wanted me to maybe try to test sort of how to do how much the picture would change with respect to I kind of imagine a scorecard is like, here's how well all the models do on rotation, and then that would then help the modelers know we need to beef up rotation, or and that sort of we can guide them in that way, right? If we really want it to be human. Again, if they want to be DARPA-like, I don't know how to guide them, but that's I think what's something we can do going forward now that we have these latent parameters under control, and that's why I like about this image generation process. As long as the computer vision community accepts that these are real tasks, and at some point if they don't, I don't know if I care, because they're really interesting, hard human tasks, and we hope that people will rally to them because we'll have a lot of neural data and psychophysical data around them. But I'm open-minded to sort of tweaks to the psychophysics, and I was talking to Ed about this last night, about how to sort of define operation in a way that's at least satisfying as a first pass of what we all mean by object recognition, because that's sort of underlying all of this, is at least taking a reasonable stab at that, and with the goal of expanding it. And that's sort of what we're doing, or trying to do, but we're open to input in those directions that would bring other people on board in terms of agreement of this is an interesting task. So thank you.